Hi, I'm Sandhya Prabhakaran and I'm here to present Sparkle, spatial reassignment of spots to cells using maximum likelihood estimation. This is joint work done with Tal Navi and Dana Pierre from Memorial Sloan Kettering Cancer Center, and I'm currently a research scientist at Moffitt Cancer Center, Florida. The research question for Sparkle. Here we find three panels A, B, and C, where A shows a smFISH image showing cells as points of green fluorescence. Panel B shows the same smFISH image as in panel A, but with the conservative cell segmentation that captures half of the transcripts. These are shown as blue dots that account for around 48% of mRNAs within the cells, and that leaves out more than half of the dangling transcripts, shown here as pink crosses from downstream analysis. In panel C, we see the spatial diversity of transcripts both within and outside the cell segmentation. Each transcript is colored according to the corresponding gene, which in the smFISH case is 140, and that is what is seen in this inset here. Sparkle aims to recover dangling transcripts from multiplexed images. The assumptions for Sparkle. The first assumption is that we do not assume a uniform distribution of transcripts within the cell. Just like we saw in the previous slide, this is an unrealistic assumption given the diversity of transcripts. Second assumption is that there's a large number of transcripts in each cell, which justifies the modeling of gene expression as a multivariate Gaussian distribution, one based on the central limit theorem, and secondly that we've checked the Gaussian assumption validity using the Lilly-Force test. Required inputs are multiplexed FISH images, imperfect region-independent cell segmentation, and the mRNA to gene mapping. Outputs from Sparkle would be improved transcript assignments to cells. It provides a more realistic per-cell quantification of each gene, a better delineation of cell boundaries, and improved cluster assignments across iterations. Some of the principles for Sparkle. One, we generate a count matrix of all cells and genes across all FOVs in order to create a global count matrix. And then one can use algorithms such as phenograph or Gaussian mixture model for clustering to identify the cell types present. Secondly, we construct a mock cell per dangling transcript. And for this, we show the same exemplar FOV that we saw before. And this is a zoomed in section of that FOV, which shows a mix of both dangling transcripts as crosses and transcripts within the cell as dots. Now, for an example dangling transcript shown here as X, Sparkle builds a mock cell where the dangling transcript is the center of the circular neighborhood, and the dimension of the mock cell is 140, that is the number of genes. There's a circular window that is drawn around X, and Sparkle quantifies the number of transcripts that are then present within the blue circular window. The mock cell is now an ordered vector, and the window size can be determined by the user. One can also create a weighted mock cell using the inverse distance weighting of each neighboring transcript to the dangling transcript, and this can be done using a modified Shepherd's distance method. Next, the maximum likelihood estimation for transcript assignment is performed. This is relatively simple and a computationally cheap strategy, which does not require customization for different imaging protocols and allows ease of parallelization. Finally, we can update the global count matrix, cluster and iterate, and in our observations, we find that 3 iterations suffice. Here is a graphical abstract for Sparkle. We begin with a set of multiplexed images. Here we see the smFISH FOVs. And we zoom in and we see the cells that are shown with transcripts that are captured within the cell segments and transcripts that are outside the cell segment. And this is a further zoomed in section to show how Sparkle creates the mock cells. And Sparkle builds a weighted mock cell for every dangling mRNA and assigns it to the closest cell having the same cluster type using the maximum likelihood estimation. And then this is then used to update the count matrix, recluster, and the process iterates. Here are some results of Sparkle. This is the recovery of transcripts across 3 Sparkle iterations, which are shown in panels A, B, and C. Sparkle captures 27%, 10.3%, and 1.2% of the total transcripts across these 3 iterations. In panel C, we can see all of the mRNA assignments from previous iterations, with the unassigned dangling mRNAs removed. Dangling mRNAs assigned to one cluster are connected to the cell centroid by lines of the cluster color, And the black dots that we see are the unassigned dangling mRNAs. Here are some more recovery statistics for the transcripts. On the left, we see the number of cells in a percentage that are recovered across the different cell types in smFISH. The percentage of cells that are seen in the paired single-cell RNA-seq data shown in orange bars, smFISH in the blue bars, and smFISH with Sparkle shown in the magenta bars. And to the right, we see the pie chart that shows the fraction of all transcripts recovered across 400 smFISH FOVs, both via segmentation, which is in blue, and by Sparkle assignment, which are these dark pink pie slices. Comparison of smFISH, both pre and post-Sparkle. Here we find the cluster-cluster cross-covariance matrices between single-cell RNA-seq and smFISH. In this matrix here, we find the cross-covariance between single-cell RNA-seq clusters and smFISH clusters before Sparkle, and this is after Sparkle. We can notice that there is a stronger within-cluster variance or a dominant diagonal after Sparkle, especially for the non-neuronal cell types. Here we find that Sparkle improves the cell type specificity of the canonical genes that are now shown on the y-axis here for most of the cell types, where the cell types are shown on the x-axis. And these are the z-scored normalized gene expressions in the rows. Next, we see the comparison of Sparkle and smFISH, both pre and post. Here we see the cluster-cluster cross-covariance matrices between single-cell RNA-seq and smFISH. In the left panel here, we find the cross-covariance between single-cell RNA-seq and smFISH pre-Sparkle, and on the right, we find the post-Sparkle cross-covariance. Once again, we find a stronger within-cluster variances after Sparkle. And we also see that Sparkle improves the cell type specificity for canonical genes in the smFISH dataset as assigned for most of the cell types. So the canonical genes are on the y-axis and the cell types are on the x-axis. So the panel of genes that was designed for smFISH was pertinent to the laminar layer of the neocortex, specifically genes like CUX2, RORB, and FOXP2. So if you look specifically at CUX2, in the pre-Sparkle setting, it was kind of spread across these three cell types. Whereas in the case of post-Sparkle, you can see that there is a very strong association of CUX2 to the L2/3 laminar layer, as can be seen here. So this is a good signal-to-noise ratio that we have post-Sparkle. Future work, future directions for Sparkle. Sparkle currently utilizes a maximum likelihood protocol, which does not assume an informative prior distribution for dangling transcripts. So our plan is to introduce a prior distribution per gene for localization of the transcripts and then implement a maximum a posteriori Bayesian inference procedure based on the spatial diversity. Another direction would be to have Sparkle in a software version that is faster, for example, like being implemented in Java. And a general issue in the field is that of cell segments, which are not really capturing the cell segments in its entirety. So if we can have methods for improved segmentation, that would help resolve dangling transcripts that are positioned farther away from the nucleus. With this, I'd like to conclude this talk. I'd like to thank the organizers for giving us this opportunity to talk at the special session at ISCB, ECCB. I also like to thank funding organizations, NIH and Python Software Foundation. Finally, this is the paper uploaded on bioRxiv. It's currently under review, and these are different ways to reach me. I really appreciate your time to having listened to this talk, and I'm happy to take questions. Thank you.